a lot of studying, but it's not a necessity to be a surgeon, but you need to do a master's of surgery if you want to be competitive in applying for surgical jobs. Every fortnight, we have a cadaver lab where we sit in front of a surgeon and they literally show us. So we went through upper limb and they just pick out every single nerve, artery, muscle, innervation, movement, and they just quiz you on the spot. And interestingly, it's actually one of the best ways to learn because when you're put on the spot, the stress that you get from getting it wrong makes you want to go home, revise, and improve on that knowledge. And I guess the good thing with me is when I'm studying, I always think about whatever I'm learning, I'm eventually going to be applying for a patient and their care. So it's a lot of gravity to it. And it's the same for all of you guys when you're doing biology now, especially when you're interested in medicine and health, you're eventually going to be using that knowledge one day to take care of another life. So it is a huge responsibility in itself. All right, I've started recording. Let me know when you all see my screen. Does anyone have any questions for Mod 7 biology before I jump into the content and quiz you all? Any questions from what you've learned? Any questions you've done during the holiday period you want to run through? Okay, good. And as you all know by now, the bare minimum is I expect you will have revised last week's content. So we will check that first. Okay, so we went through and started module 7, one of the most interesting modules, because the stuff you're learning now is the stuff that I have to apply every single day with my patients when I check up on them and see whether they've had a fever overnight. And then I need to start working out what is the causative pathogen that could be causing this patient's illness. And sometimes, scarily, guys, we never find out what the infectious cause of someone's disease is. And I have seen patients pass away in the intensive care unit because they get sicker and sicker, their blood pressure drops, their fever gets worse. And despite every single testing we've done for pathogens, we still don't isolate anything. Right. And then I have to go back to first principles and think about all of these pathogens that cause disease and ask myself, am I missing the detection of any of these agents? So what are the different kinds of pathogens that we have? Tracy, starting with you. Tell us the definition of pathogen and then tell us the smallest one you know.
So pathogens are infectious agents which are responsible for causing infectious disease.
Good.
And one of the example can be prions.
Very good. Very nice, right? I love it when I ask you guys a question and you give me an exam structured answer. Okay, because then it shows me that when you go into an exam and you have to write it down, you're going to do a perfect job at it. Okay, and that ticks away half the boxes. And then all you need to work on then is application data analysis questions to smash bio. That's all it is, right? So good. Pathogens cause infectious disease. They can be non-living, as Tracy mentioned, prions. Going to Matthew. Matthew, how do prions cause disease?
I think prions integrate themselves into the DNA, and example can be kuru, where it actually goes into the DNA and changes it from incorrect fold. I think it's incorrect folding, pretty much. Incorrect folding for prions causes disease.
Is it DNA? Have a look at this structure. Right. You see a normal prion in purple. Is it changing DNA or is it simply binding to and causing other prion proteins to misfold? It's the latter. Okay. So make sure you do not say anything about DNA, otherwise you won't get any marks. Okay. And it inherently doesn't make sense because prions aren't actually changing DNA. They're binding to proteins and causing them to misfold. Remember the domino effect that we spoke about. When one prion is misfolded, it'll bind to another prion protein and push it over like a domino into an infectious configuration. Okay, very good. And eventually, in brain tissue, all prions will misfold, and that will cause sponge-like disease of the brain. Very good. You've given us an example. I'm happy with that. That's all you need to know, guys. And maybe know one symptom of these diseases because they're so nasty. So, Sarah, what's one symptom of a prion disease?
Would it causes holes in your brain?
Yep, so it can cause hole-like lesions in the brain, so it can cause brain injury, but that's not a symptom. So this is the other thing we need to understand. A symptom is a subjective interpretation of someone's health that a patient reports. So to dumb that down for us, a symptom is like I have pain. Pain is a symptom, right? You can't measure my pain from a scale of 1 to 10 in an accurate way, right? A heroin user can say they have a pain of 2 because they've been using so much heroin, which is a pain relief medication, whereas someone who's never experienced that pain before might rate the same pain a 9. So what I'm trying to show you is pain is a subjective measurement. My 10 out of 10 could be different to your 10 out of 10. So pain is a symptom. A sign, on the other hand, everyone, is an objective measurement. If I measure your temperature and it's 38 degrees, it is 38 degrees. It doesn't matter if we're talking about your temperature in winter or summer or how much pain you're in, temperature is always going to be the same quantitative number. So that's the difference between symptoms and signs. So when I ask for symptoms, signs, I don't want you to tell me the disease process. I want you to tell me what a patient might come in with. Okay. So one symptom you might like to mention is features of dementia. So memory impairment, difficulty speaking, difficulties in movement. Ask yourself, what does the brain do? The brain does everything you are doing right now. Moving my hands, speaking, thinking, memorizing, all of those compartments and functions of the brain deteriorate. Good. That's everything about prions. That's all the detail you will need to know for your exams. So let's move on to the next disease. Alyssa, what is the next smallest pathogen?
Viruses?
Very good. Viruses living and non-living?
Non-living.
Yeah, good. They're mostly regarded as non-living. Some can consider them living because they have genetic material, but that leads us to our next question. Kriti, what is the general structure of a virus?
There's like the DNA in the inside, and the outside has like a I forgot what it is. A protein?
Good. It's a protein capsid. Okay. Protein capsid. Mohammed, next. What's outside of this? What's outside of the protein, Mohammed?
Envelope.
Good. So envelope made out of lipid or fat. You need to understand this, right? Rather than just memorizing. I can tell we're memorizing when we don't understand and remember the different parts. DNA or RNA is extremely fragile. If I picked your DNA out and I put it outside of the nucleus, it will get destroyed in seconds by enzymes. Right? That's why mRNA needs a uracil. Uracil is like an armor coat to the genetic material so it can survive on its journey to the ribosome. So all viruses logically need to have a thick protein capsule, which we call the capsid, which protects that genetic material. The other thing that we've said is DNA. It's not just DNA. A lot of viruses, in fact, are RNA. Most viruses are RNA, some are DNA. Okay, so all you need to say for your exams is genetic material. So be careful how you word the structure of a virus. And finally, they have a lipid envelope. Now, can you hit the raise hand icon? Have you seen who's seen Harry Potter? Any of the movies? Cloak of invisibility. That's what a lipid envelope does. Your cell membranes are made up of a phospholipid bilayer. When a virus has a lipid envelope, it allows the virus to kind of merge with your lipid envelope of your cells and enter inside. That's what the lipid envelope does. It's like a cloak of invisibility. It protects the virus from being detected as foreign by your immune cells. When you understand this stuff, it's easier to recall the structure of these pathogens. Okay, good. How does a virus cause disease? I think Matthew kind of mixed viruses and prions. Viruses have a role in causing disease through modifying DNA, etc. But let's go to Sarah. How does a virus cause disease? Sarah, viruses, disease?
So basically, it disrupts the function of the cell.
Good, good. But again, I need us to have an answer that will give us full marks in an exam, and that wouldn't be it. Good overall picture, right? It does disrupt functions of a cell, but do you agree that's quite vague? Everything disrupts the function of a cell. If I change the pH of a cell, it's going to disrupt the function of a cell. Who can tell me an exam answer? How does a virus cause disease? Do you all agree? I'm asking you very simple questions. Your exams aren't just going to ask you, describe how viruses cause disease. These are all, I would say, the bare minimum content you need to understand before we go through exam content questions. Okay. Alyssa, on to you. How does a virus cause disease?
It injects its genetic material into another cell, so like into a host cell, and that cell then replicates the genetic material, and then that's how it spreads to other parts of other cells.
Very good, right? So now we've got a whole additional layer of information that Liz has given us, which is that this genetic material of the virus is going to enter into a host cell. The only other information I would have wanted to have seen is this is a universal principle. I think those of you who are in the chem class would understand this. Whenever you learn something in biochem physics where there is overlapping content from different modules, it is extremely high yield. One of the overlapping bits of information right here is DNA replication and polypeptide synthesis. If you do not say those two words when you're explaining how a virus works, just you have not answered the question. Okay, and you will not get the marks. A virus enters into a host cell and it's going to replicate its DNA or its RNA using the host cell polymerase, ligase, primase, etc. The main enzyme being polymerase that I want to hear in your exam answer. Okay. What does a virus also have? You've all told me a virus is also made up of a protein capsid. That protein capsid is going to be made from getting this viral RNA and running it through a ribosome in the host cell to make the proteins. So in summary, a virus causes disease by binding to host cell receptors, entering into a host cell, using polymerase enzymes of the host cell to replicate viral genetic material, and using ribosomes and tRNA molecules to synthesize viral proteins that will finally assemble and then burst out of the host cell, killing it in the process. That is how viruses cause disease. Do you guys see the level of detail that we're at and where we need to be? Okay, very good. Just make sure before you come into class, you've gone through this and without looking, you explain it all to yourself. Only then can you progress towards, fine tuning and smashing the bio exams. Good. Let's move on to Kriti. Give us an example of a viral disease, Kriti.
Influenza.
Very good, right? And Trissy, can you give us one more example? It's kind of here, but any others that you know in addition to influenza and HIV, Trissy?
Maybe AIDS.
Yeah, so AIDS is actually, it's the end stage of HIV. So when someone has a very, very low immune cell count, or they have a really nasty disease because of HIV, we classify them as having AIDS. AIDS stands for Acquired Immunodeficiency Syndrome. Acquired immunodeficiency. You don't need to memorize this, but I'm just telling you the relationship between HIV and AIDS. So they're actually the same disease, just different stages of illness. Okay, good. Any other viruses that we know? There's so many. The common cold, influenza, COVID-19, of course. Ebola. Who remembers Ebola? You would have all been, I don't know how old you are now. If you're 18 now, you would have been maybe 11 or 12. I don't know how old you guys were in 2016, but that's when Ebola persisted in West Africa. and we were scared it would be a pandemic. Ebola caused what's called hemorrhagic fever. Hemorrhage means bleeding. It'd give you fever, but you'd also start bleeding out of all your mucosal orifices, the eyes, the nose, the mouth, the gastrointestinal system. It was a very infectious and very nasty viral disease. Good. Okay, now this is the part where we have to start talking about plants, because biology is about life in general, right? When it comes to plants, what you need to know for the HSC is the pattern of disease that you would expect from the different kinds of pathogens. So how would a virus cause a plant disease? How would a bacteria cause a plant disease? How would a protozoa or a fungi look if it caused plant disease? And for your trials, you should know one example of each pathogen. So you should know one example of a bacterial plant pathogen, one example of a viral plant pathogen, one example of a fungal plant pathogen. It's not that much, just 3 examples and then 3 patterns of disease, and you're set for all of plants. Okay? Now here's my tip to you. Who's seen plants like this? When I was in high school, I remember I used to just pick interesting looking leaves or flowers and just have a look at them. Has anyone seen a leaf that looks like this? Good, right? We've been perceptive. You've all seen this. This is the most common pattern of disease, and virtually all plants have plant disease. When a virus affects a plant, it causes a mosaic pattern on the plants. Well, what does the word mosaic mean? Does anyone know what the word mosaic means? All of you should, because you learned about the fluid mosaic model of the cell membrane. Tracy, what is a mosaic pattern?
I think I first heard this word in art, but it's kind of like the scattered like patterns of like
Very
glasses
good.
and stuff.
Very good, right? So you often see that on churches in depictions of Christ and God. You'll see the panes in different colors. And I think that's for many religions. They have their, I think it's also in Orthodox churches, you'll see patterns of glass in various different colors. Same thing with the fluid mosaic model of the cell membrane. There's a pattern to it. So mosaic is just a pattern and usually refers to a pattern of colors. So that's what you see here. Mosaic disease in the leaf. You see a pattern where there's scattered bits of different color in the leaf, some lighter, some darker. So this is mosaic disease. So remember this, if you see discoloration, right, in a plant, typically is a viral disease. Next time you look at a plant, just have a look and you will see this. 100% I can guarantee you, if you just walk in the park outside, you will see one plant that has this disease. And that's telling you it's being devastated currently by a virus. Very good. Here are a few examples. Pick any example. I just pick rose mosaic and I just use rose plants. Okay. That's the disease. This is the actual virus, the prunus ring spot virus. But you can possibly just remember apple mosaic virus affecting roses. I'm sure it also affects apple plants. But, just remember mosaic disease caused by a family of mosaic viruses that can affect plants and apples. If you say that in your exam, you will get more than full marks. The HSC will not test you on this. I need you all to appreciate this, right? This bit of just brute memorizing stuff is very trials content related. In the HSC, they will test you much more on the things I've asked you. Okay? I'll show you an example of a HSC question if I can find one. Right, if I just go down here to Bio 7 trial questions, scroll through. What did I tell you? It took me literally 5 seconds to find a question, and you can see why I stressed why you need to know the structure of these pathogens, because they'll test you literally based on what a pathogen looks like. The other kinds of questions you get in your trials and your HSC, they're all application-based questions. So quickly do this question for me. I'm going to get Mohammed, you're going to tell me what pathogen A is. Kriti, you're going to tell me what pathogen B is. So you're working at the microbiome lab at it could be Prince of Wales, RPA, Liverpool Hospital, and I'm calling you urgently because the patient is dying from this bug. This is what you see under the microscope. Tell me, what is the pathogen? I've called microbio asking them because we had a patient in ICU who was extremely unwell and I needed the results ASAP so we could start this really severe last line antibiotic because the patient was extremely unwell. And this is exactly what they did. They did PCR of the genetic material of the pathogen. They tried to culture the pathogen in different mediums, and they looked for toxins that the pathogen made as well. What is pathogen A? Mohammed?
Prion.
Easy, right? So you actually don't see prions under a microscope. Prions are something we only see, it's usually post-mortem after someone dies. The coroner, who's a doctor that looks at, dead bodies after they die to find the cause of death, will look at it under a microscope and realize, this isn't a typical pathogen. And then they'll eventually look at it under an electron microscope, etc. They'll see the prion proteins, and that will tell us there is prion disease. Okay, pathogen B. Let's go to Kriti. What is pathogen B? Undergoing binary fission and it's prokaryotic. What is this?
Bacteria.
Very good, right? Typically, when someone is really sick, I take blood cultures where I take a sample of their blood and I grow it on a petri dish, which is a nutrient-rich agar medium. The lab does that for me, and then they'll call and tell me what the bug is, and that allows us to start antibiotics if it's a bacteria. Very good. So the answer is clearly prion bacteria B. 3 marks if you know the structure of a pathogen. Good. Let's come back up here. All right, and that leads us to bacteria. What I need you to understand is even though you're learning all these different families of causative organisms, the 2 highest yield are viruses and bacteria. 90% of patients in hospital with an infectious disease have a bacterial infection or a viral infection. Okay, if I was to see something like a protozoa or a fungi, I would think something else is up with their immune system. Either they have a weakened or immunosuppressed state, that could be due to various causes, or they have really severe illness. I usually never see fungi or protozoa. So bacteria are very, very common. So just like we learned from the previous question, they are all single-celled. They are all prokaryotic. What are the other features of a bacteria that you must be able to recognize? It's all written in front of you, but let me ask you, Matthew, what is a cell wall of a bacteria made out of?
Isn't like chitin or something? Trident?
That's fungi.
Cellulose?
Not plants.
I don't know.
Can anyone tell me what's the cell wall of a bacteria made out of? One thing, guys, if you struggle with the remembering stuff, you need to first identify that. And then my tip is use flashcards. You can use an app called Anki, which is what we do in medical school because the content just goes up exponentially in university. Use a flashcard app, or if you really want to, make physical flashcards yourself based on things you continually forget. There's no such thing as having poor memory. All of you have good memory. It's just you haven't dedicated the time that you individually need to remember that topic. Okay, so I hope you're all finding out what you're weak at in terms of whether it's application questions, whether it's remembering the core content, etc., and your study technique reflects that. Okay, good. If you want, message me and I will tell you, because I've taught you guys for a while now. I know what each and every one of your weaker areas is, and that will help you be more proactive in the holiday period. Let's go to Mohammed. Mohammed, any idea what the cell wall of a bacteria is made out of? You might not have been here when I first taught it to everyone, Mohammed, but
Is it the salt with phloem?
no, that's plants. We're getting further from the answer. Bacteria. So you need to understand this. The word cell wall is just a wall that is outside of the cytoplasm and the cell membrane. Beyond that, the cell wall of a fungi is completely different to the cell wall of a bacteria, which is completely different to the cell wall of archaea, which is another example of a prokaryotic organism. Atul, if your mic's working, what is the cell wall made out of? Your mic is very good. Peptidoglycan. Good. And for those of you who are wondering what the heck peptidoglycan is, here's my tip. When you learn a new word, break it down. Peptido is telling you there are peptide bonds, so it's protein. Glycan is telling you there's glucose. So a bacterial cell wall is made out of amino acids and sugars, or protein and glucose. As long as you say that, you'll get full marks for your exams. This is so important, everyone, because all antibiotics, that 99% of them, work by binding to the cell wall of a bacteria and destroying it. Humans don't have a cell wall, hence why antibiotics do not affect us to the same degree. Very important to understand. Okay, good. What other features do we have for a bacteria? Let's go to Sarah. Can you tell us what a pilus or a flagellum is?
Is the flagellum is it's what absorbs the water?
Close, kind of. Not. It doesn't absorb water. Flagellum is a motor. It's like a boat, right? Think about it. Your body is 60% water. Bacteria need to travel through this water. So this is kind of like their motor that allows them to turn and push and propagate through your body. This is how they move. What about pili? Let's go to Trissy. Any idea what these pili do? What are pili?
Does it help it stick onto the thing?
Very good, right? So think about, let's give you an example of pneumonia. Pneumonia is a bacteria that infects the alveoli or the air sacs of your lungs. So patients with pneumonia have chest pain because it's in their lungs, it's not just in their throat. They have shortness of breath, they get fevers, and patients die because of pneumonia. It's a nasty disease. The bacteria adheres onto your air sacs using these pili to attach and take hold. So even if you cough, they stay, they don't translocate and they don't get coughed out. So pili are very important for attaching to the epithelium of your cells, which are the lining cells of your airways, of your gastrointestinal tract. Okay, very good. Other things to note, all of you should tell me they have one circular loop of chromosome, and you should always draw it as such. Good. That's all I can say. They only have one organelle. They have ribosomes, and that's because they're prokaryotic. Only ribosomes are non-membrane bound. That is also a super high bit of information to know. Good. How do they replicate? This is mod 5 revision knowledge, binary fission. Nothing else to say. What are plasmids? You learnt this in mod 6. So do you see how the more you learn, the more things just start to get categorized? Because this is the second or third time you're learning it again. So Matthew, what's a plasmid?
It's pretty much DNA within the prokaryotic organisms.
Yep. Is it
Like
is it the same as chromosomal DNA or is it a different sequence?
no, it's a different sequence.
Very good. So what I would say in an exam is there's small circular loops of DNA that are independent to the chromosomal DNA, and they can be transferred horizontally to other organisms, right? So I can't just pull out my genes. I can't just pull out my genes for memory, etc., and throw it to you, right? Whereas bacteria can literally pull out genes for adherence, translocation, toxin production, and then just throw it to their buddy bacteria, who will then take it in and start making the protein or the toxin. That's how bacteria evolve so quickly, and that's why resistance is such a problem. If we use an antibiotic to kill a bacteria, some bacteria will just inherently be resistant. They'll be born with a thick cell wall, or they'll be born with a very small pores in their cell membrane that prevent the antibiotic from actually reaching the bacteria, and those bacteria will replicate. And remember, their lifespan is like 20 minutes. They'll replicate and make millions within, a couple of hours and become resistant to an antibiotic. Right now, there are bacteria that are resistant to every single antibiotic we know. And that's because certain countries have misused and overused antibiotics. And that's why we say don't use antibiotics unless you truly have a bacterial illness and you need to. Okay? Antibiotics, you need to be very careful because resistance, right now, if you get infected with a bacteria that is resistant to everything, you will just die. There's nothing we can do. Even if you come to hospital, we can just give you medications to increase your blood pressure, slow down your fever, try giving you some weird antibiotic, which will have a lot of side effects, but it won't be too effective. Good. All right. Two very simple points to understand. You understand how a prion causes disease, domino effect, it misfolds other prions. Sorry, my iPad randomly turns its audio on as well and echoes. Just give me one sec. Let me go back here. Good. So prions domino effect, they misfold other prions, cause disease. Viruses invade into cells and use DNA replication, protein synthesis machinery. Bacteria cause disease in two ways. They will use their flagella, ramp up that motor, and ram into the cell lining and invade through tissue, destroying it in the process. Kind of like a submarine missile, right? Spin their motor and they'll just enter in and penetrate tissue, or they will secrete toxins, any chemical that will disrupt normal cellular function. Those are the two ways in which bacteria cause disease. Urinary tract infections are a very common example of a disease, and I'll show you what happens. 90% of urinary tract infections are caused by E. coli. You might have heard of this bacteria. This is the bacteria that we use to make human insulin, and we transgenically modify it to make other things for us. But E. coli, it's a rod-like bacterium. And what it does is it enters in to the urethra or the urinary system. And if this is an epithelium or the lining cells of a urinary tract, it will take itself and ram itself in and invade through tissue. And in the process, that will cause injury to the cells. They'll release chemicals that will recruit immune cells and start the inflammation process. And you'll start to feel pain when you pass urine. Very rarely, there may be some bleeding when you pass urine because of this invasion as well. So you'll get frequency, you'll get the sensation that you need to pee a lot, you'll get urgency, you suddenly need to go to the bathroom, you'll get pain, and that's typically pain over your lower abdomen, right over the pubic bone. Those are all the symptoms of a UTI that's caused by this bacteria. So it's just a little case example, okay? Another one is secretion of a toxin. Now, everyone has had gastroenteritis, which is the disease where usually it's a virus that infects the gastrointestinal tract and it causes nausea, vomiting, diarrhea, etc. Right? Now, the way that certain bugs cause this gastroenteritis, let's just break that word down because it will teach you a lot about disease. Gastroenteritis. This is the most important word I want you to understand because as a layperson, when you see this word, you will see it eventually because all of you will get some kind of itis in your life. It could be hopefully not meningitis, but you can get hepatitis, you can get gastroenteritis, colitis. Does anyone know what the word itis means? Any ideas? Itis means inflammation. Inflammation for now, just understand as redness, swelling, etc. So gastro is telling you it's a stomach. Enterite or enteritis or the enteric system is the small bowel. So the stomach and the intestines are inflamed. That's what this is telling you. Okay. Now, in the case of Shigella, what this bacteria does is it goes and sits in your gastrointestinal tract. Remember, this is your mouth, and then it's your esophagus, and you have your stomach, and then you have your small bowel. Just think of it as one long tube from your mouth all the way to the rectum and the anus, right? That is the gastrointestinal tract. This bacteria will sit here and it will secrete toxins, these chemicals. And what the chemicals will do is they will draw a lot of water due to osmosis, right? Because remember, if there's more toxin, there is now a lower water concentration, and water wants to move from a higher water concentration to a lower concentration. So via osmosis, it travels into the gastrointestinal tract, and then you end up with diarrhea. That is a mechanism of diarrhea for some of these agents. So good. So this is just 2 case studies. You don't need to memorize this. But as long as now you understand what you mean when you say invasion into tissue and secretion of toxins. Does anyone have any questions about how bacteria cause disease? Good. All right. When it comes to picking examples of bacterial diseases, pick either very common ones that you will see as a layperson or you will see as a healthcare worker in your future, or pick really interesting ones that truly fascinate you. Don't just pick a random bacterial disease. I think some really interesting ones. Meningococcal disease. Can you hit the raise hand icon? Have you heard of meningococcal disease? Okay. What I want you guys to search meningococcal. Just be warned, you might see some graphic images, but it won't be too graphic. It'll just be bruising over someone's skin. Meningococcal. Actually, let me search it first, just so it's not too confronting. Oh, it's not too bad. Oh, I guess it's very relative. But yeah, can you guys search meningococcal? Just search meningococcal for me. And don't go too down because there are some bad images. But just go to the first image or the second image. You'll see a boy which has this discoloration over their skin. All right, this isn't a rare disease, by the way. It is not a rare disease at all. What I'm showing you here is meningitis. Meningitis. What is meningitis? This is your brain. Do it in pink. This is the brain. And the brain has these tissues, these layers of tissue that coat it inside the skull. Right, so this is the skull, the brain, and this layer of tissue that provides nutrients, blood flow, and protects the brain is called the meninges. Meninges. When you get inflammation of the meninges, it is called meningitis. Remember, itis is inflammation. Now, when this inflammation is due to a bacteria, we call it bacterial meningitis. That makes sense. Meningococcal disease is a specific type of bacterial meningitis that is caused by the bacteria Neisseria meningitis. Okay? Now, this bacteria is really nasty because it doesn't just cause a meningitis. It releases toxins that causes bleeding from blood vessels underneath the tissue of the skin. And so what you saw in those images is bleeding or bruising underneath the skin because of this bacteria. So if I ever see a patient in the emergency department and they have a headache, neck stiffness, fevers, I start thinking meningitis. One of my friends in high school had meningitis, so it's quite common. You might know someone who's had meningitis before. It's definitely, and it's very common among your age group, which is another very interesting point. Teenagers, young adults, just due to being in close proximity, it's transferred through droplets through the nose and the mouth, can cause meningitis to others. And so the lining of the brain can swell, and that can be very deadly. And very quickly what can happen is it can cause permanent brain damage, hearing problems, permanent intellectual disability, especially if it's not treated with antibiotics, and especially the meningococcal disease. If you saw the bleeding under the tissue, which is that bruising you saw in the children, can cause rapid, massive bleeding, impair blood flow to organs and tissues, and eventually cause limb amputations. And this can happen in a span of 24 to 48 hours. A normal, healthy teenager can get fever, meningitis, get this bacteria, and then end up in the intensive care unit needing an amputation. And I've seen cases of this, so it's not a rare thing at all. If you literally just search meningococcal New South Wales Health, you'll see there's stories of teenagers who talk about their story with meningococcal disease. All of you, I think, are vaccinated against this, but that's just a very interesting disease, but also a really nasty disease to know about. Meningococcal disease caused by Neisseria bacteria. I don't think you'll need to have to say the full names. If you can just remember Neisseria, that's more than enough. Does that make sense? Does anyone have any questions? I definitely said a lot, and there's certain words that you might want clarification on, or mechanism of disease. Any questions so far? Okay, good. Another interesting one is tuberculosis. Tuberculosis is caused by a bacteria, Mycobacterium tuberculosis. And this bacteria goes and it affects the lung tissue. Very contagious disease, very contagious. And it goes and causes massive hole-like lesions in the lung tissue. And it can spread in the bloodstream and cause death as well. If you have tuberculosis, you need to be on antibiotics for 6 to 12 months, up to a year. And the other scary thing is you will need to be permanently isolated in hospital. You're not allowed to come in contact with the public until you are de-escalated, aka you're not contagious anymore. It's a very contagious disease. So if anyone has tuberculosis or TB, which is the short for tuberculosis, we'll need to put them in a negative pressure room, meaning air can never get out of that room. Air only goes into that room. We'll need to put them on antibiotics, and I'll need to notify that condition to public health so that they can track that case of tuberculosis and see if it's spreading anywhere. So it's another nasty disease. You see it a lot in South Africa. So if any of you do medical school and you do an elective term in South Africa, you'll see a lot of tuberculosis with HIV. And they kind of work in conjunction. HIV destroys the immune system, and that weakened immune system allows tuberculosis to take hold and cause severe infection. Is anyone from South Asia? So it could be anywhere in Asia in terms of ethnicity. If you are, then tuberculosis is very common in those countries. So a lot of immigrant workers who come to Australia or Sydney from overseas, especially Asia, they can actually have tuberculosis sitting in their lungs very quietly, right? And their immune system could be controlling it for now, but then under the stress of working, moving places, not eating too well, the tuberculosis can reactivate and cause massive disease in the lungs and kill them. So we see a lot of overseas workers who come here get really sick with tuberculosis suddenly and then pass away. And that's because of this case. Good. One symptom I always ask patients that screens for tuberculosis is this. If they've traveled from another country and they're coughing up blood and they've got weight loss, then I would not be a good doctor if I did not test for tuberculosis. Okay? Does anyone know how we'd test for tuberculosis? Who's anyone done module 7? It's not a fancy test, it's a very logical test. And it's something that I've mentioned earlier as well, to test for bacteria. What do we do in hospital to test for bacteria, guys? Starts with a C. Mentioned it a couple of times. Culture using a petri dish. So you can get any body sample. If it's tuberculosis affecting the lungs, we take a body sample from their lungs. So we get them to cough up some phlegm. And then we put that phlegm and we wipe it on a petri dish and we send it to the lab. And then if the lab grows tuberculosis, then we have to immediately put this patient in a negative pressure room and make sure that you literally need to wear pretty much a hazmat suit whenever you interact with them. But that's tuberculosis for you. Okay, so the other key point here is for bacteria, the main way we identify them is by culturing the bacteria, aka growing it on a petri dish. Can you hit the raise hand icon? How many of you have seen a petri dish or grown something on a petri dish? Maybe in a previous school report. Very good. So you guys literally know how to grow a bacteria. That's exactly what we do in the hospital setting, but we just grow it on different kinds of nutrients to help us isolate and grow different kinds of bugs. Very good. So I would say pneumonia, tuberculosis, meningitis, bacterial meningitis are some really interesting bacterial diseases. You can also pick urinary tract infections. Those are extremely common. I can guarantee you. In fact, a woman, just because the urethra is very close to the rectum anatomically, much more common to get urinary tract infections. So most women can get, up to 1 to 3 urinary tract infections a year, and that can be completely normal. So in terms of commonality, very common. UTIs are urinary tract infections. Good. All right, we'll briefly talk about gram staining. Has anyone heard of the word gram staining before? No? That's all right. So gram staining is a very quick way to categorize bacteria that we've grown. So let's say that, you've got a patient who's unwell. Where's my pen not working? So you've got a patient that's unwell and you're concerned about tuberculosis. You've got their sputum or their phlegm sample and you're looking at it under a microscope. You'll often see little... Sorry, give me one moment, guys. My pen doesn't seem to be working. Give me one sec. I'll get you guys to quickly do a question. I'm just gonna get my pen to work. Just using my hand now. Let's do this question in the meantime. Oh, these questions are insane. "2019 Sydney boys. Give this question a go." Hit the raise hand icon once you've got an answer. Okay, I'm just gonna restart my Teams, my iPad. Give me 1 sec. Might be an iPad thing. Good. All right, what do we get as our answer to that question? Did anyone get an answer? Good. Atul, can you type your answer? And Mohammed, what was your answer?
I got fungus.
Fungi. Good. So if it's heterotrophic, I think they said heterotrophic cell wall or eukaryotic, any of those features would cue you into it being a fungi. Very good. Good. All right. I think what we'll do is we'll take a 5-minute break. By then I'll figure out how to get this pen to rework. And then we'll touch upon the rest of bacteria. We've pretty much finished it. It's just the gram staining is a very low yield small point I want to mention. It's because we often, when we get a sample of a bacteria, before we culture it, because remember, culturing is when you grow something in a petri dish and you're trying to see whether you can get microscopic bacteria to grow in such big colonies that you see it with your visible eye. That takes days. Culture results take 1 to 2 days. To very quickly help us identify what kind of bacteria is in a sample, we can stain the sample. That's what gram staining is about. We'll talk about that, and then we'll probably finish the different kinds of pathogens, and we'll start talking about disease transmission. Okay? Take your break. We'll recap at about 2:05. Okay? See you guys. All right, when you're all back, hit the raise hand icon. Nice. Cool, we've already done this question. Nice, let's go back. Do any of you have a teacher at school who's really good? Doesn't have to be a subject that you you like per se. Do any of you have a teacher at school where they explain things in a very simple, easy to understand manner, or they show you what's interesting about the subject, or they seem passionate about it? Any of you? You hit the raise hand icon if it is the case. Good. Trissy, what subject does that teacher teach?
English.
Nice, nice. And do you like English as a subject?
Or after he taught us.
Yeah, it's very interesting, and it's important to realize this. A lot of who you will become as an adult is really dependent on who showed you how interesting a certain topic is. That's just really it, right? I think every topic, from literature to science to history to engineering to computer science, is all very fascinating and interesting. It's just about who shows you the beauty of that subject. And it's very similar even in medicine, because there's so many different specialties. Where someone ends up in terms of their specialty is actually largely determined by how their experience with the team or with this, the boss surgeons, being involved in teaching, understanding things, is versus the actual topic itself. Just keep an eye out to that. A lot of where you might end up might be because someone showed you how interesting computer science is, for example, or how interesting maths is, and you end up doing it in university, and that becomes your whole career. And sometimes if you feel like there is a topic that's interesting, but you're not being taught it well, I would encourage you to go seek out that help. Find someone who can teach it to you well, so you don't miss out on that entire career just because you have a bad teacher. Good. Good. All right. Let's come back to this. Gram staining. Okay, so like I said, how long does culturing take again? Let's go to Sarah. If I had to grow a bacteria on a petri dish, how long would it take before I can see a visible growth with my naked eye, Sarah?
It takes a few days for it to grow.
Very good, right? It's very important to realize that when you send a culture off, you need to be checking for that in 1 to 2 days. That's what I need to do whenever I send off blood cultures or sputum cultures or stool cultures for my patients. But that takes a long time, and someone could be really sick, and you might need to treat them before those culture results come back. We can't wait till someone ends up in the ICU or their fever gets so bad that their blood pressure drops and they get, they almost die. So at that point, we need to start treatment early. And gram staining helps us do that. Because what gram staining does is as soon as we have a sample, whether it be blood, whether it be the phlegm, whether it be any body fluid, we can look at it under a microscope after staining the bacteria. And we can stain the bacteria using this purple dye. And the way it works is that bacteria with a very thick cell wall will retain the purple dye that we stained. The way you think about this is if they have a very thick cell wall, when we stain the bacteria with that purple dye, it stays and accumulates in the bacteria, but the cell wall is too thick for the dye to come back out. Whereas with bacteria that have a thin cell wall, the second you stain them, the cell wall is so thin that it can't actually hold in all of that dye. And all of that purple dye comes right back out, and the bacteria will end up having a pink appearance or a very lightly stained appearance under a microscope. And that's what you see here. When I look under a microscope and I see this, everyone, I know this is gram positive, aka a thick cell-walled bacteria, because it's retained that dye. Whereas when I look under a microscope and if I see this, right, with very faint pink splotches, which represents every single bacteria, then I think, okay, this is a gram-negative or a thin cell-walled bacteria. And this gives me an answer in minutes as to what the bacteria could be. Okay, so that's what gram staining is all about. We stain bacteria with this purple dye, and thick cell-walled bacteria appear gram-positive or dark purple, gram-negative appear pink. There's a little bit more to it, to the method of gram staining, etc., but that's all way outside of our scope right now. Any questions about gram staining? TLDR, if you forget all of this, it gives you a very quick answer as to what the bacteria could be, because there are very specific organisms that have a thick cell wall. There are very specific organisms that have a thin cell wall. So let's say a woman comes in and she's complaining of mild fevers, some burning when passing urine. I get a urine sample and I run it through a microscope, and within an hour, the lab calls me back and says, I've got gram-negative rods under the microscope. I can straight away start antibiotic treatment for E. coli bacteria because I know it's only E. coli which would give off this appearance. And now I don't have to wait 2 days for the urine to be cultured and grow E. coli in macroscopic or visible colonies. Kriti, did you have a question?
What does it like mean when it's like gram positive versus gram negative? Like, what's the difference?
Yeah, so again, the difference is just the cell wall thickness, Kriti. So remember, gram-positive bacteria, they have a thick cell wall. So they'll retain that purple dye and they have a purple appearance. So when something is gram-positive, I just know the bacteria has a thick cell wall, and there are very specific bacteria that have a thick cell wall. So, if you're doing medicine, you'll know there's bacteria called staphylococci, there's bacteria called streptococci that have a gram-positive cell wall. So when I see gram-positive organisms grown, I think about these guys. Whereas if I see a gram-negative bacteria grown, I straight away think this bacteria has a thin cell wall that was so thin it couldn't retain that purple dye. And so now I start thinking about E. coli if I sent off a urine source. And there's all these other weird kind of bugs which we don't need to know that are also gram-negative that I would be worried about and I'd need to exclude. So these are all just different bacterial species. Does that answer your question? Purple, thick cell wall bacteria, pink, or gram-negative, thin cell wall bacteria.
Yeah.
Any other questions, guys? Again, this is low yield stuff. It probably won't come up in your exams, but if it does, they'll briefly talk to you about the gram staining method, or they'll tell you about gram staining and then give you a sample and ask you what it could be. And that's just applying your knowledge. Trissy, question?
I was just wondering, like, if antibiotics, like, they are specific to certain bacteria, and you test it, like the bacteria has like gram like negative, won't it be like not precise enough for the bacteria?
Could you repeat that question? So you said antibiotics are very sensitive. What was the last part of your question again?
I was just wondering if like antibiotics, they are like really specific to bacteria, it's like specific bacteria. So if you test it for gram negative and you give a patient like an antibiotic, wouldn't it be like not precise if it's like another bacteria?
Yes, very, very good point, as Tracy mentioned. So like I said, there are groups of bacteria that have a gram-negative appearance, and there are multiple bacteria that have a gram-positive appearance. So just based on the gram stain, Tracy, I can't start a narrow spectrum antibiotic or an antibiotic that acts very specifically on one bug, because I still don't know exactly what bug it is. Right? So in that case, I would start an antibiotic that has a broader spectrum of activity, meaning it acts on a wider range of bacteria. So not all antibiotics act on only one bacteria. We have different antibiotics, some that act on all bacteria, but don't act really, really well on any one particular bacteria. And we have certain antibiotics that work on very few or one bacterial species, but work really, really well. And the goal is we don't want to use broad spectrum antibiotics ever in medicine, because using a broad spectrum antibiotic would mean we're killing the bad bacteria, but we're also killing the good bacteria that make up your gut microbes, that make up your entire microbiota. And that can cause a lot of side effects like diarrhea, etc., because we're killing good bacteria as well that control bowel motions and all of those things. Does that answer your question? So until the culture result comes back, I will start them on a broader spectrum antibiotic. And when I finally find out which one of these three bacteria it is, for example, I will then switch to a very narrow activity antibiotic. Good. Very good question. Any other questions at all? Good. So let's go to Matthew. Matthew, what do we need to know about plant infections again? What did I say for your exams?
Actually don't remember at all.
Sorry, what was that?
I don't remember.
You need to write it down because we just spoke about this, right? Alyssa, what did I say we need to know about plant? Oh, just plant infections or the different kinds of plant infections for your exams.
Isn't it that we needed like one of each?
Good. One example. And then a very brief outline of the pattern of infection. Can you all write this down? Everyone who didn't get it the time. So plant bacterial infections, you need to know one example and the pattern of infection. So only two things you need to know, right? So tick it off as we go through the different plant bacterial infections. So we went through viruses. Kriti, what kind of pattern of infection do viruses have on plants? What do they cause?
The mosaic patterns.
Very good, right? So they cause this discoloration of the leaf that's darker in certain parts and lighter in certain parts. Very good, right? Now with plant bacterial infections, they cause disease very similar to human bacterial infections. Now, in a human bacterial infection, we typically get a lot of inflammation, swelling, we see a lot of redness, and we also see discharge, right? An example is, for example, acne. Acne is caused by bacteria that grow under blocked skin follicles. Now, acne is not always just caused by bacteria. A lot of the time, there's hormonal factors at play, etc. But the end result is inflammation happens because bacteria grow inside the follicle, and that can cause a discharge or a pus, which is what we see, which is the accumulation of dead white cells and bacteria, right? So plant bacteria have a similar pattern of infection. They can cause an ooze, and that ooze is also known as discharge. That's a medical term for ooze that comes out of a wound. There's discharge. Okay? Discharge is the word. Good. They can also, bacteria in plants specifically, they can release certain toxins or chemicals that can increase the rate of mitosis in a plant. This is specifically only for plants. This doesn't happen in humans. So they can release toxins that increase mitosis, and that can cause overgrowths or swelling of the plant. And you can see that happening here. You can see there's a bacteria here that's inserting its genetic material. It's acting kind of like a virus. And you don't see that with human bacteria, hence why I don't want you to really remember this. But just understand they release certain hormones and chemicals that cause parts of a plant to start dividing rapidly to form an overgrowth. And if you've ever seen a plant that looks like this, where it's got these tumors on its surface, tumors are masses, right? Those are very characteristic of bacterial plant infections. And that's called gals. I need you all to say these words, otherwise you're not going to remember it at all. So gal is a swelling or overgrowth on a plant that happens because of bacterial infection. Discharge is another sign of bacterial infection. And the last one is blight. And the way I remember blight is blight has a B. B for browning of leaves. So the leaves on a plant will turn black/brown. And unlike them turning light green. So if you look up there, where was it? You can see that viral diseases, they cause, a light green discoloration of the plant, which is very different to the blight or the browning of a leaf that I'm talking about. So these are the three signs of a bacterial plant infection. Okay, very good. Now, one example you can remember of a plant disease, I remember this because Can anyone tell me? Where did this word ring a bell? Agrobacterium. Where have you seen this word before? Anyone? So this is a bacteria that inserts the Bt toxin gene. It's a vector that inserts that Bt toxin gene into cotton plants. Cotton plant embryos are dipped in a solution of agrobacterium that will grow, that will transfer the Bt toxin gene. So that's how I remember the agrobacterium. And in this case, the agrobacterium causes gall or overgrowths in a leaf. Okay, Agrobacterium crown gall. Make a palm card out of this. I would say this kind of stuff is something that's just hard to, it's not really understanding but memorizing. It's good to know for your exams. I'll give you an example. Let's have a look. See how specific they are? I can tell you 80% of students that sat that year's paper that saw this question did not know the answer just because they didn't remember an example. And again, like I'm telling you, this doesn't come up in the HSC. This comes up in your trials. They ask questions like this in your trials. They don't care how well you memorize stuff in the HSC. When you do the HSC, you realize it's a different paper altogether. It's much more application based. So those of you who are good at applying the knowledge you've learned, I'm sure all of you are good. You just need to fine tune it. Will do well on that kind of paper. Whereas your trials is just solely who knows the most plus who can apply the best. Good. All right. Oh, that's topic. Come up here. Good. Any questions about bacteria? All right. Protozoa. Trissy, question?
I was wondering because some like deficiency, like nutritional deficiency of plant could also cause like some appearance of the plant. So maybe like for example, like a deficiency of like potassium could cause like yellow patches on the leaves. So how do you distinguish between like disease causing leaves or like deficiency causing leaves?
Very good question. The answer is I would not be able to. I'm not a botanist and you are not required to either. So I'll never ever give you a question where you see discoloration on a plant and then have to distinguish it from a nutritional deficiency, which is a non-infectious disease, to a, for example, mosaicism, etc. For me, how would I distinguish them? It's very hard, especially with a plant, because, wilting of leaves, um, discoloration, they're all very non-specific signs. And it's the same with human infections as well. I can't look at an infection and 100% always say this is bacterial, this is viral. Sometimes you do need to send off, microbiology samples to get a unanimous answer as to whether there is a bacteria that's growing on the petri dish or whether there is a virus that we've detected from PCR testing of the body fluid. So to answer your question, I think even botanists would struggle to answer that. Some things that would help would be if you understood what that mineral or that metal did for the plant, and then you could work logically backwards and try identifying what the features would be. But for your level and even for my level, we don't need to know the differences. But it's a very good question. Good. Any other questions at all? Question shows me you guys are thinking critically rather than rote learning. So I love to hear questions. Whenever I was learning this, I had a ton of questions. And that's what I'm here for, right? Okay. Protozoa. Let's go to Sarah. What does a protozoa look like, Sarah? If I look at it under a microscope, what would tell me that there is a protozoa?
Is it a 3D structure?
They're all 3D, right? There's no 2D structures. They're all 3D. Bacteria, viruses, every living organism, everything outside of my iPad screen is 3D. Maybe you mean 3D like as in a globular structure. That's more proteins, but can anyone tell me? Give you a case example, right? So a teenager or a university student comes back from a trip to South Asia and they've now got fever, they've got dark urine, they've got really bad headache, and they tell me that they went to a country where a lot of mosquitoes are present and they weren't able to use repellent. And what I do is I sent off a sample of their blood. It's a test called a thick and thin smear. So we get blood, sample of blood, and we get a thick smear to look at it. And that tells us how much pathogen is present. And then we get another sample of blood and we grow a very thin smear being a very light sample of blood to identify the causative agent. So it's a thick and thin smear. You don't have to memorize this stuff. What would I find here that would tell me this is a protozoa when I look under a microscope at this blood sample? Anyone? Gritty? What does a protozoa look like?
Well, it doesn't have a cell wall, so it just have the cell membrane.
Very good. That's one feature. Is it eukaryotic or prokaryotic?
Eukaryotic.
Very good. The only prokaryote, Matthew, is what organism in terms of pathogens?
You said they're only eukaryotic?
Yep, protozoa are eukaryotic, but do you know what pathogen is prokaryotic? There is only one pathogen that is prokaryotic. It's also unicellular. We already went through it today.
Bacteria?
Very good. So bacteria are prokaryotic. Okay, protozoa are eukaryotic. So they're eukaryotic, unicellular, no cell wall. That's it. Protozoa, eukaryotic, unicellular, no cell wall. You can literally make Anki cards out of this, right? And it would help you understand it very easily. So whatever you struggle to remember, just consider making flashcards. It'll help you a lot. Good. So you're completely right. Under a microscope, I'd expect to see this. It's eukaryotic because I can see the endoplasmic reticulum. I can see the mitochondria. It's only one cell. Now, can anyone tell me why was I suspecting a protozoa? What do you think this patient had? Fever, headache, dark urine, confusion. Very interesting. The fever in this condition, it comes up and down. There's patterns to the fever. If it happens every 48 hours, I'm concerned about a different type of bug compared to if it happens every 72 hours, etc. What am I worried about here? Mosquito, fever, what disease? Trissy?
Malaria?
Very good. This is textbook like features of malaria. And you aren't required to know the symptoms and signs of malaria, but all I wanted you to identify is if I say the word mosquito, an infectious disease, you say malaria back. Because if you can do that, you know everything you need to know for malaria for now. Okay? Very good. Malaria is caused by a protozoa that sits in the digestive system of a very specific mosquito called the Anopheles mosquito. So this is a mosquito digestive system. This is a back piece. Okay, so here we have the protozoan, and the name of this protozoan is the Plasmodium protozoan. That's the genus name. There are multiple different kinds of Plasmodium protozoa. Okay, the most common one, or the nastiest one, is Plasmodium falciparum. There's also Plasmodium vivax, ovale, malariae. Do you need to know these? No, not at all. Control Alt Delete from your memory. But as long as you understand Plasmodium is the name of the protozoa, that's all you need to know for your exams. Plasmodium protozoa which is the causative organism for malaria. How do we test it? We look at it under a microscope. Symptoms, as long as you remember fever, that's more than enough. Mohammed, question?
Is proteus and
So you cut off for a sec. I think your question was, is protist and protozoa the same? Is that your question?
yes, that's
Yeah, so protists are a kingdom. Protozoa refers to protists that can cause disease. That's the difference. But they're often used pretty interchangeably. But protist is like saying the word animal, plant, fungi. They're kingdoms of living organisms. So protist is anything that is unicellular and eukaryotic. Not all protists cause disease. Protozoa refers to protists that cause disease. Very good. So the main important protozoa you all need to remember is malaria, caused by the Plasmodium protozoan. There's a few others. The only other time I'm concerned about a protozoal infection in patients is if they start getting nausea, vomiting, diarrhea. Who remembers what the name of that condition is when you get inflammation of the stomach and small intestines? You should all know this because all of you are going to have this in the future as well. Gastro is a short word, and gastroenteritis is a long word. And the layperson word is food poisoning. When you say you have food poisoning, you're saying that you have gastroenteritis that's caused by some kind of bug. Okay, sometimes gastro or food poisoning can be caused by protozoa. In 1999, there was a huge outbreak of a protozoa in Sydney from its water source that caused a really bad gastro, and everyone had to boil their water on a cooking stove. That's what I was told when I was back in high school. Good. So malaria is the one I want you to remember. So the one I was talking about is Giardiasis, and there's also Cryptosporidium, which are two different protozoa that both cause protozoal gastroinfections. Do you need to remember this? Not at all. Just malaria. If you guys can do that for me, then you're set for protozoa. Okay, so you should tell me what a protozoa looks like. How does it cause infection? The exact same way as bacteria, so nothing to say there. And then give me one example. You're set for your exams. You just really need to know the structure well. As you can see, it took me 30 seconds to run through past papers and find 3 different questions testing you the structure of the different pathogens. That's probably the most high yield part. Okay. Look at how weird they look. This is them under an electron microscope. This is a Giardia protozoa. This is one cell, but look how funky that cell looks. Right? You can see the multiple flagella. It looks like a jellyfish, but it'd be floating around in the digestive system. And these are its eggs that it releases. Nasty. But the test I send off when I look for this is a stool OCP. So if a patient has diarrhea, it's really bad, and I don't know what the cause is, to be safe, you send off stool OCP, which stands for oocyte, which are little baby parasites, cysts, which are kind of like these eggs that are dormant, and parasites. Again, this is just additional info. You don't have to remember this part, but just how it's relevant to doctors. Any questions so far? Plant diseases with protozoa. I would just say, just read this. Don't even remember this. I would say this is so low yield. I've never in my 7 years seen a question asking you about a protozoal plant infection. Extremely low yield. So just read this and know that they can cause plant infections, but don't commit any of this to memory. You can see, because similar to bacteria, they can also cause blight. So you can think of them very similar to bacteria in the way they cause disease as well. Good. Any questions so far? All right. Maybe let's do another question because we've done a lot of talking. Let's do some good questions. Here is a good question. This is a tough application one. You'll have a minute to answer this one. A minute and a bit. Okay. Read it carefully. And this is testing more your scientific technique. And like I told you, Petri dishes are high yield because this is how we grow bacteria. All right. Who's got an answer? Hit the raise hand icon. Good. All right. Let's go through it. Trissy, don't worry about your answer. Actually, Trissy's answered so many questions, so I'm gonna pick. All of you have done well. Matthew, what's the definition of validity?
Pretty much whether a method tests what it's intended to test.
Good. So validity is, did you meet your aim? That's the first question you ask yourself. And how do we check if an experiment has met its aim? Alyssa, what does an experiment need to do to meet its aim? Sorry, I just put the volume up because I can't hear too well. Alyssa, could you repeat that? Alyssa, any ideas? What?
Hello?
Yeah. So what part of an experiment tells you that it's met its aim?
If it's got like controls and independent and dependent variables.
Very good, right? Does it have a clear independent, dependent variable? Now, you have to be very careful when you say this. Control and control variables. They're two different things. What's the difference, Kriti, between a control and a control variable?
I'm not entirely sure, but I think controlled variables, they're kept the same throughout, but then a constant is just one you do at the beginning to like, I don't know.
Okay, so you guys need to be masters of accuracy, validity, reliability, because this stuff will probably predominate multiple marks in your HSC exam. Okay, so again, independent is what you change, dependent is what you measure. Yeah, let's find out what is it independent, what is it dependent for this experiment. I want someone to tell me. So all of you should have been doing this when you're asked a validity question. If you go straight to guessing an answer or just, off the top of your head, you'll have a lot of holes in your reasoning that very well could stuff you up in your final answer. So Sarah, what's the independent experiment here? What's the dependent experiment? And you can take a bit of time to re-skim it if you need. So I'll help you out here. 6 students prepared 10 petri dishes using sterile techniques. Okay, so we've got 10 petri dishes. 8 were exposed and the other 2 left unopened. So there's 2 petri dishes that were never opened. Okay, and there are 8 that were left exposed. I'm guessing that means that were left open for the air. So we've got 8 of those. Okay, they're all placed in a location for 20 minutes. So this is 20 minutes in a location at the same time of the day. So we're controlling all these variables. And then they were incubated, which is when you put them in a warm 37 degrees little oven so the bacteria can grow into macrocolonies. The total number of bacteria and fungi were grown and counted. So Sarah, what's the independent variable?
The location of the dish, because it was the same, and the time of the day, it was the same.
Those are control variables. What's the independent variable? The independent is what you change. What did we change between the 2 petri dishes?
One of them was left open and one of them were left closed.
Good. Exposed or open petri dishes. So what we changed was exposure of the petri dishes to the surrounding air. Everyone with me? Exposed, open to air. They weren't swiped with anything. There was no rod that swiped the surface of the petri dishes. They just said it was exposed. And what are we measuring after a couple of days? Let's go to Mohammed. Total bacteria. So what do we need to control? What are some other variables that can affect the total bacteria that will grow on that petri dish, the total bacteria and fungi that we controlled? Alyssa? What are some variables that were controlled that have been mentioned already?
The time that the petri dishes were left.
Good. Time of day,
Incubate?
duration. Yeah, I can hear you. So time of day, duration of exposure. Very good. Okay, good. So we can clearly see the experiment now. And now my question is, what is the control? Matthew, what was the control for this experiment? I'm not asking for control variables. What is the control?
Control is the ones that are left closed.
Very good. So the way you answer this, everyone, is the control variable is testing whether there really even is a relationship between your independent and dependent. That is the question that the control replicate or the control trial is doing. And what we do is we remove the independent variable and we see whether it affects the dependent variable. Does that make sense to all of us? Okay. So you remove the independent variable. So in this case, we'd have a petri dish that isn't exposed. Okay, there's no independent variable, and we measure the total bacteria and fungi. And if we see that removing the independent variable still causes a big change in the dependent variable, then we know the experiment isn't valid because it must not be the independent which is affecting the dependent, but other factors that we haven't measured in our experiment. So again, to summarize, control, we remove the independent variable and we measure the dependent. And what we hope to see is that when there is no independent, there is also no dependent variable. If there is a dependent variable, then it's telling us some other factor that we haven't considered is contributing to growth of bacteria. Okay. And then if that's the case, we're not actually testing whether exposure to air is causing bacterial growth. We're also testing how these unknown other factors, which could be not using aseptic technique or not growing the petri dish in a sterile manner, could be contributing to growth. So now, given that you understand all this information, you're experts in validity, whose experiment was valid? So Atools has an answer. So can you type your answer? Kriti, did you have an answer? And if so, what was your answer? Anyone have an answer? Good. Matthew, what was your answer?
A.
Okay. They all were valid because he's used a control. I think that's a reasonable answer for now. Atul said it could be D. Tracy, what do you think?
I think it's D as well because the true
Okay.
variable has no
Yeah,
bacteria.
good. So, look, I understand why Matthew picked A because to be valid, you need to use a control, but you also need to control variables, which were both done here. But again, the only issue that I said, this is the most important part of validity, is the question, did you meet your aim? Now, for everyone you can see except Ali, even when the plate was never opened, there was still bacteria growing. AK, when it was never ever exposed to the air, bacteria were growing. So that's telling us that the total bacteria and fungi for a lot of these cases, have a look at Nick's moldy bread, right? He must have exposed that petri dish to moldy bread. He must have swiped the petri dish with moldy bread. At least half of the growth was not even due to the moldy bread. It was due to some other factor he didn't control, which he doesn't know. If you go and ask Nick what caused the unopened petri dishes that were meant to be sterile to grow half a load of bacteria, he would have no clue. Right? And so he hasn't met his aim because every time he's doing his experimental result, at least half of that is coming from some other unknown cause. AK, even though he used a control, there was some part of his method where he didn't control some variable which is causing this growth of bacteria. So in summary, just because an experiment has a control and has controlled variables, or they've said they've controlled some variables, doesn't mean it's valid. Only when they've controlled all variables, and you can see a clear relationship where if you remove the independent in your control, there is no dependent, can you say the experiment is valid? So in that case, only Ali's experiment is valid. And I'd agree with Trissy and Atul that D is the answer here. Okay. Everyone else, this question's trying to fool you. They're telling you, oh, they were placed in a particular location for the same time of day. Sure, they controlled those variables, and sure, they said it was sterile technique, but then what's causing all these bacteria to grow? Okay, a small growth would have been okay, but having, literal half the growth of the experimental result in a control that was never ever exposed to any bacteria is poor form. There is some external variable that we're not controlling that is causing the growth of bacteria. And to have a valid experiment, you need to identify those variables and you need to control for them somehow, which we have not done, except for Ali. Ali had a valid experiment. D is your answer. Okay, hopefully you guys learned from this question. I thought this was a really good question to do. All right, coming back to our actual content here. All right, we're talking about fungi. Let's go back to Mohammed. Fungi, tell us a bit about the structure, Mohammed. What does a fungi look like? Mohammed, quickly, what does a fungi look like?
Biotic cell wall.
Good. What's the cell wall made out of?
Chitin.
Good. Chitin, it's eukaryotic, which means it's got a nucleus with chromosomes, with DNA. Outside, it would have an endoplasmic reticulum or some other kind of membrane-bound organelle. There would be mitochondria, and there would be a cell wall made out of chitin. So the main way, everyone, that you distinguish between a protozoa and a unicellular fungi is the cell wall. So that's why I'm really stressing for you to remember this. Cell wall eukaryotic. Do they have to be unicellular or can they be multicellular, Kriti? Fungi. What's your final answer?
I mean, they're like classified under unicellular, right? So,
Mushrooms. I hate mushrooms, but they're a perfect example of a fungi that are multicellular. So your answer is multicellular. They can be both. Okay, so that's another differentiating factor. Fungi can be unicellular or they can be multicellular. Alyssa, when a fungi is multicellular, what would its method of reproduction be?
Sexual spores.
Very good. Yeah, so it'd be sexual, can sometimes be asexual spore. So just spore formation is the answer. Whereas Sarah, if a fungi is unicellular, what is the most likely method of reproduction?
Is it binary fusion?
Not binary fission, that's bacteria. What's the next most complex method?
Budding.
Very good. Guys, this is also very high yield because you learnt it in mod 5 as well. Okay, so bacteria is binary fission, protozoa is binary fission budding, fungi is budding and spore formation. That's how you remember it. Okay, every more progressively complex life form takes one method of reproduction from the previous life form and adds one additional method of reproduction. Very good. Other things to mention about fungi in terms of fungal diseases. Fungal diseases tend to affect the skin, and that's because fungi very commonly occupy our skin. Right now on your skin, we have fungi. Okay, but they're kept in low quantities because they're fighting the bacteria on the surface of your skin for resources and nutrients. It's literally a battlefield right now in your skin, your hand. The times where fungi start to outgrow the other normal bacteria is when your immune system gets weakened. So a perfect example of this is thrush. Do not search this up. It's quite graphic, but I will describe it to you, and maybe at your own discretion you can look at it if you'd want. Thrush is a fungi that grows in the mouth or in the genital tract. Okay, so it can grow at the in the vaginal wall. It can grow in the mouth and the tongue, and it only overgrows when someone is immunosuppressed. So some key examples are, do we have any asthmatics here? Anyone who's got asthma? You're all amazing lung genetics. Nice. But in asthmatics, we usually give them a steroid inhaler to suppress the inflammation in the lungs that asthma can cause. And a side effect of inhaling steroids is steroids suppress the immune system. And so if you don't wash your mouth after, taking the puffs of the steroid puffer, you can start developing thrush on your tongue, which looks like a cottage cheese-like discharge that's coating the tongue. Similarly, in patients who have diabetes, which is a form of immunosuppression, or HIV, or even pregnant women, it's very common for women in pregnancy because the body's spending so much resources on the baby that the immune system becomes a little bit weakened. They can also develop vaginal thrush. And it's very easy to treat it. You just use antifungal creams or oral antifungal mouthwashes for thrush. So that's a very common example of a disease. And we're going to come back to thrush, everyone, because it's an example of a disease that only comes on when the immune system is weakened. And that's another dot point we'll need to understand. Other examples of fungal infections, most of them are skin-based. Ringworm. You can search ringworm up. Can you all search it up on your phone? Ringworm's a beautiful example of a disease that you can spot diagnose, and it looks really cool. When someone walks in and they've got a bit of itchiness on their back, and you see these ring lesions, and without even doing any tests, you tell them, I know what you have. It's a fungal ringworm infection. So search ringworm up for me. And hit the raise hand icon once you've got it up. So ringworm is not caused by a worm. It's a misnomer, right? It's not caused by a worm. It's caused by a fungus, which is, as you know, a unicellular, eukaryotic, sometimes multicellular organism. In our case, certain fungi can cause these lesions. As you can see, they have this target or ring-like appearance where the center is clear and the periphery of that lesion is circular and red. And again, you can treat that with antifungal creams. That can happen in the body, it can happen on the back, it can happen sometimes on the scalp, ringworm infections. The last point I'm going to leave us with is with plant infections. Virtually all plant infections are fungal. So although we spoke about protozoa, which cause maybe 0.01% of plant infections, although we spoke about bacteria, which can cause maybe 10% of plant infections, 85% of plant infections are fungi. You don't need to know the stat, as long as you remember, virtually every single plant infection is fungal. And there are a few patterns of infection that tell you that it's a fungi affecting the plant. One such example is rust. I want you guys to search this up, plant rust. I'm sure you've seen this very commonly. The brown, it's not a brown, it's more like a bronze discoloration of leaves. So just search rust plants. And it looks like the rust of metals, right? This orangey color in a plant. That is a spot feature of a fungal plant infection. Right? I've seen it so much. It looks like I'm looking at the school playground again when I look at these Google images. Virtually every single plant has this. Another feature is mildew. Mildew is this white powder that forms on the surface of a plant, and that's just reproducing fungi and their structures that give off this white powdery texture. So mildew and rust are two patterns of infection in a plant that tells you it's fungal. Okay, good. Now, if you forget, you don't have to remember every single example of a plant disease and its causative agent. For your child, if you can remember one, it would be eucalyptus rust that's caused by the Puccinia fungus, Puccinia species of fungi. And this is an example of an Australian plant and the fungi or the pathogen that affects it. This is extremely hard because this is a dot point in your syllabus, which you'll come across later, which is whether you can name an Australian plant and the specific pathogen that causes it. Puccinia fungi that causes eucalyptus rust. Okay, good. So if you forget every single named example of a pathogen and causative organism, this should probably take you through 90% of those questions. Good. All right. Very good. Any questions? Nice, good. I want you guys to do these questions. I wrote these questions up for you all to do. Make sure that's done by next week. Sorry, I think there has been a delay with postages with a long weekend. But if you haven't got your booklet yet, you'll definitely get it this week in terms of doing the questions on paper, okay? And I'll make sure you guys all have the Google Drive access as well. I just need to go through, have another chance to double check and get you all access. But this is homework, and I will, by tonight, you'll all get online access for the notebooks as well. Okay, good. Any questions at all? Nice. All right, good job, guys. Enjoy your long weekend. Try doing some stuff outside of study. Keep yourself sane, balance yourself. This year isn't all about studying, so try to still catch up with friends, play some sport. Do whatever you're passionate about. I think the biggest thing that I would have changed if I was back in year 12 was I'd be more self-directed with the things I liked. So if I genuinely was interested in coding or engineering, start learning a language. The very cool part about today's age is a lot of jobs don't even require a degree, and they just require you to know the knowledge. In the case of coding, in the case of being a doctor or a lawyer, you need to get, university credentials. But for certain jobs, even like commerce, that a lot of people go into, um, university helps a lot. But I think doing a lot of self-directed things, learning coding, or even, doing any kind of extracurricular stuff along that career trajectory will help you a lot as well. All right. See you guys next week. Bye.